From Microbe TV, this is Immune, episode number 36, recorded on October 20th, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi. I'm back in my office, at least for today.
You look very professional today with plaques and diplomas and theses. Those are all theses behind you. I know it, right?
No,
They're
those
lab
are
notebooks?
lab notebooks.
Oh, I thought some of them look like theses. Some people have.
Some might be. They're old notebooks.
Also joining us from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. I do not have any lab notebooks or theses behind me, but lots of things for teaching classes.
Yeah, I do have a thesis. This is my thesis. Look at this. 1980.
Wow.
1980.
Wow.
But the cool
Right.
thing, it's a lot of pages. It's 300 plus, and it's typed.
Whoa.
It's typed by hand. I had to hire someone to type it because we didn't have computers. And every figure is a print that was cut out and glued in, and press-on letters. I don't know if any of you are old enough to remember that. Probably not. You're all too young.
I had to do that early on.
So I keep my thesis here next to me, and this is all where it all started, guys.
Oh, very nice. When I was in grad school, we all used to have a contest to see if we could write a thesis long enough so that our name would be written horizontally instead of vertically. It looks like you won the contest.
It was horizontal. This was, by the way, about influenza B and C viruses. And the thing is, I had to write a draft by hand, and then I had someone type the draft because my advisor would not read handwriting, and he corrected it, and then it was retyped. Ridiculous. And to this day, I don't edit my students' theses because of that experience, because
I'm guessing that it was done more than a few days before your thesis defense.
Oh, yeah, it was done a long time before. I spent hours
Students
sitting
these
at my
days they'll get me their thesis like 3 or 4 days before the defense.
They give
They're
it to
supposed
you.
to be there 2 weeks
They
before.
don't even get a week. And I spent hours at the dining room table pasting those pictures in. There was glue, that kind of rubbery glue. Anyway,
Yep.
Well, Steph is off doing something. So let us introduce our guest today. He's from Indiana University School of Medicine. He's a professor of microbiology and immunology. Alexander Dent. Welcome to Immune, Alex.
Well, thank you.
Can we call you Alex?
Please. Yeah. That's what
Great.
I go by.
All right. And apparently,
It's good to have you here.
Yes,
it's very good to
much
have
so.
you here. What's it like in Indianapolis? Is it cold yet? It's winter? No, not yet.
Yeah, it's like mid-50s today in the day. So, yeah, it's not too bad.
We're having a 70-ish day today, right, Brienne?
Yes, we are.
It's incredible. It's just
really
gorgeous.
nice out there.
It got cold and now it's warm again.
It's pouring down raining in Ithaca.
Really?
Ooh.
Wow.
Yeah.
Well, it could
all
be
day.
snow. It could be snow,
That is true.
Hey, look at that head.
Hi. Yes. So one of the things
Oh my
that
gosh.
we have during COVID times is, and this is very relatable to the pandemic. So there was an infant whose parent tested positive, and the infant was also positive. That was in Graham's daycare class. And so what that means is I don't have childcare.
Wow.
But I did not want to cancel because we had this on the calendar. So I think
thank you.
I can do a little multitasking. And so I'm here. You might see me come in and out, but I figure in terms of podcasts and science, this is kind of like the reality. You're balancing family, you're balancing a pandemic. So I'll be here in and out, but I'm really thankful that, and is it, you prefer Alex? Is that
Yes.
great? I'm glad that you could come on. You're active on Twitter. I've read your papers, and the paper we're going to talk about today is very relatable to what you study. And I thought you'd be the perfect
I love it.
person.
Yeah.
So we thank Graham for coming on as well.
Yes.
Yes, we do.
He'll do little guest shots.
In 20
I
in
just
20
wanna grab
years,
his cheeks.
Yeah, we do.
in 20 years, you can show him this video and he'll go, Mom, I had my son on episode 7 or 8 of TWIV, right?
Did
He
you?
was all enthusiastic, and now he's kind of embarrassed whenever I mention it. But people love it. He talked about viruses in video games, and people loved it. But he's now totally embarrassed. So, Just wait, Steph. Just wait.
I know. I can control him now. He just sits here.
So you guys were not tested, right? You and Graham and...
Well, what I did was I was able to quickly get a test. He had ironically had a pediatrician appointment that day. And so I had mentioned this and they ordered up a test. There was only one testing site in Durham because I'm Duke Health that did infant swabbing, and he is PCR negative. Now, of course, if the audience is a listener of TWIV, we know that maybe one PCR test isn't always conclusive.
Right. That's right.
But we've had no symptoms, no other indications. So I think we're okay, but we do still he needs to quarantine for those 14 days.
Yeah. Well, hopefully it'll be all well and you'll be well as well. So
but
it will go by in no time.
Sorry to interrupt.
Go ahead.
I was going to say my daughter is in high school and she had a classmate who tested positive. But my daughter wasn't listed as a close contact, so
really
she wasn't ordered to quarantine.
Huh. That's interesting.
Yeah.
I would have thought that in the classrooms, if they had one that's considered close contact.
I don't know. They're kind of weird about it.
Interesting.
They say like you have to be within 6 feet for 15 minutes. That's their CDC guidelines.
I find that funny.
It is funny, isn't it? Look at your watch. 14 minutes. Let's go.
Time to go. Right.
All right. Yeah. So we have a great paper today. But before we do that, Alex, just give us a rundown on your background. I think you're from Los Angeles initially, right?
Right.
Born and raised there.
Right. Well, I was born in Northern California, but basically grew up in Los Angeles. Grew up in North Hollywood, Studio City, if anyone knows that area. And then I went to UCLA for undergraduate and got interested in immunology there. And then I went to UCSD for my PhD, where I worked with Steve Hedrick on T-cell receptor and thymus development, and I worked on gamma delta T cells. I was fortunate enough to get one of the first gamma delta T cell clones that we cloned out the T cell receptor genes for and made transgenic mice, and got a nice paper on that to look at development of those cells in the thymus. And then I went to NIH for my postdoc with Lou Stout, who does B-cell lymphoma work and some immunology, but he's more of a transcription factor. Now he does all this. He pioneered like microarray analysis for tumors, for particularly B-cell lymphoma, and he's developed a lot of RNA interference techniques and that sort of thing. So he's now he's a National Academy member. So I haven't talked to him for a long time, but he's obviously a great scientist. And I've been here 20 years now. Time flies. It's crazy.
So what building were you in when you were at NIH?
10.
Yeah. Okay. I was too.
Yeah.
When did you leave? So it must have, I don't know if we overlapped.
98. Yeah.
98. No, I came in 2000. We just missed each other.
So it's interesting. Lou Stout was a postdoc with David Baltimore, as I was. And let me ask you, at UCLA, who taught you immunology? Did Owen Whitty by any chance?
No. William Clark was a guy I worked with, and also Eli Sirkars, who you probably remember. He died.
I do.
A few years ago. Then there was a guy named Ben Bonavita who worked on NK cells that I worked with for a little bit. Okay.
It would have been interesting if you had worked with Owen Whitty, because he was also a Baltimore postdoc, and you would have
right. Yeah, I think he was. I knew of him, but I never actually interacted with him at all.
Yeah, we actually overlapped in the Baltimore lab, and to me, he was notable because he would come in the morning and he had this piece of paper in his shirt pocket with all the things he had to do that day, and he would check them off as he did it and walk around the hall putting this back, and I hated him for that so much because he was so organized. Well, I didn't hate him, but I kind of felt inadequate.
I haven't heard his name for ages. He's like famous for like B-cell developmental cultures, right?
Yeah, well, he's the first guy to find that ABL was a tyrosine kinase. and started that whole thing. But yeah, she's done well too. So what
Maybe
you?
wet cultures or something like that? Didn't he do something?
Yes, that's correct.
Okay.
That's correct. So you never wanted to go to work in Hollywood, huh?
No,
No,
no, I was never interested in acting.
no, because usually you grow up in L.A., you want to do that.
I did want to be a punk rock star for a little while.
Didn't work out.
I guess.
Well, from what there's also a YouTube video I think people could look up if they wanted to, but are you also a ballroom dancer? Is that true?
Yeah, that's been my more recent hobby. Yeah,
Okay. Wow. So that's pretty cool.
Yeah.
yeah, it's a lot of fun. I love it.
Yeah, that's great. Well, it seems like there's a lot of connections. I think science, particularly when you get into subfields, it's quite small.
You bet. Small world. Yep.
Yeah, small world.
You bet.
Oh yeah.
All right, Steph. As long as you can drive
Yeah,
the paper
sure.
discussion, take it away. It's a cool paper.
Great. It is very cool. And this paper, firstly, I'll read the title. I'll read the authors. It's quite the long list of authors, so I won't read all of them, but the title is Loss of BCL6 Expressing T Follicular Helper Cells and Germinal Centers in COVID-19. So the first author I'll read is Naoki Kaneko, and the last author is Shiv Pillai. And it's particularly the Massachusetts Consortium on Pathogen Readiness Specimen Working Group, which I don't know if that is a particular acronym that spells COVID. It seems to be there's a lot of working groups that are developing where the acronyms spell COVID or some type of cute name. Right. But
I think there are 4 co-first authors.
Oh, thank you. Yes. And actually I'm going down. Thank you for pointing that out. Let's give everyone credit, because if you're like the first co-author,
And no one ever talks about you.
No, that's not cool. Right.
And they're not alphabetical. So
No, they're not. So let's see how I do here. So I'm going to say the H is silent. So Sal Suan Kuao, Julie Bukau, and Jocelyn R. Farmer. That's the first 4. And those are the, first co-authors. I think in these large collaborative groups, you have a lot of people doing work that can be considered, equally contributable. So
Absolutely.
yes. So we got to make sure we give credit to where credit is due. And so this paper is really interesting and exciting for a couple of reasons, but what they wanted to identify was why, firstly, you have these differences in people and their ability to respond to SARS-CoV-2. You have a large disparate response in immunity, and a lot of it's driven, what we think, by mostly the immune response. And so we know that because particularly during the viral phase of infection, you may have an increase in viral titers, but that does decrease over time. And then you have the second phase of innate immune response. And actually, I know, look at me having to go. So who wants to take over for me as I go into the situation?
I don't know what you were going to say exactly, but yeah. So I can start a little bit. So one of the things was we've done a paper before that was talking about the limited immune response in certain patients, right? And so I think the key part of this particular paper, which is that if you don't have appropriate T follicular helper cell activity, then you don't get appropriate B cell responses and productive antibody and class switch responses in the germinal centers. So that's a whole lot of words that we talked about. T follicular helper cells are not cells that we've talked about much before. But germinal centers we've mentioned, and those are really the nidus, basically in a lymph node or in the spleen, where cells congregate to be activated. So B cells primarily will be educated near there and then proliferate and expand and mature and class switch and produce antibodies. So all that really happens in the germinal center, but they need help. And they get that help primarily from those follicular T helper cells. I think that's where she was going to start. So.
Yeah. So I think the other thing that's really important to point out about that is that in the germinal center, the B cell does the class switch, does somatic hypermutation, and gets help from follicular helper T cells. But that also allows B cells to undergo sort of the programming and differentiation that might be needed to make a good memory B cell response.
Yes.
And so those germinal centers are the places where B cells get educated into memory B cells. And they talk here a bit about how there may be short duration of antibody responses in response to this coronavirus, some other coronaviruses, and they even talk a little bit about some other viral infections in general. And that they had seen, and they knew that those antibody responses are initiated in secondary lymphoid organs. And they said, well, we've seen the secondary lymphoid organs look a little strange in some autopsy samples from people with viral infection. And so they wanted to sort of look in more depth at the secondary lymphoid organs to see if there were changes that could be linked to those aberrant responses. And so ideally, they want to link the duration of the antibody response and memory with the structures of the secondary lymphoid organs, with the T follicular helper cells, like Cindy mentioned, and sort of they want to be able to see if they can put all these things together.
Yeah, and I think the major advance of this is that in prior studies, they've been sampling peripheral blood.
Exactly.
Right. And a lot of these cells, and we've critiqued those before, you can't find many of these populations circulating because they home to the lymph node or germinal center, and that's where they park and they don't really leave. And so if you're sampling peripheral blood, you're missing a lot of what's going on. And so the power of what they've done here is they've been able to get these autopsy samples from severely ill patients that were hospitalized, and they stratified them into, I guess, very severe and semi-very severe based on their blood levels of some inflammatory markers.
I think they said the early group was people who very early, and then the late where they took longer to succumb.
Yes, they had 8 days. If they died within 8 days of, I think, the onset of their symptoms, then they called them early. And if it was 15 to 36, they called them late.
Yeah. So I think do we want to move into the figure one? We're sort of getting close to it with that part of the conversation.
Did you have any more background that you wanted to talk about? I think one of the main things we want to say here at the outset is why is it important to study this? It's important to study this. I think from one of the very last things they write in the paper is that suggesting that natural infection may not be the best way to generate immunity, right? Because if you're not able to generate the memory T cells and the memory B cells and the long-lasting durable immunity, then it may not be as protective. So not that our vaccines are all that great at inducing those responses, but they may be better, or we may adapt them so they could do this. So I think that was one of the takeaways that's really important. The other one that's really important is that this particular viral infection is basically wiping out the differentiation of one particular subset of cells in the immune system that's really critically important for, all these downstream effects that eventually lead to host protection.
Yeah, I think one caveat of this study is they're looking at people who died from this disease who had very severe inflammation. So that kind of colors the whole analysis. But the results are very striking for sure. So I mean, because it's a little hard to know how much you can extrapolate to people who have severe disease and then survive versus people who die from this. But,
right.
Yeah, it's a really interesting paper.
Yeah, I think it's an important point because we'll come back at the end. The wiping out of germinal centers may not happen in people who recover. So what's their antibody response like? Is it more durable? Is it more somatically hypermutated as a consequence? That's an interesting issue, right?
Yeah, exactly.
Okay.
Yeah, I think that's a key point.
And there's a lot. Sorry to interrupt, but there's so much controversy about this durability of the antibody responses, and I always feel like people never put it into enough context because you see the antibodies go down, but antibodies are always going to go down.
Absolutely.
After an acute infection, and is it going down below like detectable levels, or is it there's still some low level we don't. It's never really compared to anything else. So.
Yeah. The other
and
thing.
I was
Go ahead.
I always wonder about the fact that we're still not that far out from most of these people's first infection. And so our ability to say that the duration is not great or that the antibodies have waned is really something we're measuring in a pretty short time scale.
Yeah.
If we look later, we could get some other interesting things.
Yeah, the other
popping back on. I've been following along this whole time, so I'm up to date with what you're talking about. But yeah, and it's interesting because I think, a lot of the conversation about durability obviously is in some of these studies that have come out to show that the antibodies don't last that long. And why is that? But it's a lot of retrospective studies looking at common cold coronaviruses and seeing that similarly, those antibodies are waning. They're not something that's durable. And it's really centered around the germinal center reaction, which was one of the reasons I thought Alex coming on would be great, because this is a really important concept when we're thinking about immunological memory and developing something called high affinity antibodies. And so the name high affinity really is just saying that the affinity of that antibody to the antigen is strong and that it has undergone positive selection in the germinal center over multiple rounds to get to that point. And so obviously what they wanted to do, and I like what Cindy said, having this ability to look at something that's not PBMCs or peripheral blood mononuclear cells is important. And they show that here in this first figure. They show you by doing some very cool imaging. I've not really done imaging like this where you're using multiple colors, and they're looking at it at like the single cell level. I mean, they're trying to quantitate by microscopy, which I think we could talk about the validity of that over something like flow cytometry. But what they overall show you is that in COVID-19 patients, both early and late, there is a really drastic lack of germinal centers. And overall, a decrease in B and T cells, which we kind of know about the virus, something called lymphopenia. But I mean, look, and these images, I don't know if someone wants to share their screen of these images because it's a very visual paper, but it's quite striking.
Yeah, they're beautiful images.
Yeah, so I find this really striking because there's sort of this major loss of B and T cells. And to my eye, one of the first things I see when I look at figure 1B is where are the T cells. There certainly, I can say, yes, there's a loss of B cells, but the absolute lack of T cells is quite striking to me. And the nice part about doing this with microscopy is that they can also actually look at the architecture of the organ, which is so important for a secondary lymphoid organ like the lymph node. And in this case, they're looking at the thoracic lymph node, which is the lymph node that should be draining the respiratory system. So it's the lymph node making the local responses.
Yeah, and I had a question for Alex. When you look at germinal centers, or we're going to talk about something called BCL6 expression, do you have a particular lymph node that you look at? I think for our audience, it's interesting to know that the lymph node you choose is very important in the infection.
Yeah, sure. I mean, typically, if you want to see a response, you would look in the so-called draining lymph node, which is where the antigen's going into, and that depends on where you immunize. A lot of times we look at the spleen. That's the easiest thing because a lot of responses eventually accumulate in the spleen anyways. So it really depends on the model we're using. But like mesenteric lymph nodes, we look at a lot.
Yeah, which are in the gut. And so, like, as Brienne said, thoracic might be a good one to look at because it's draining from the respiratory tract, and you can be able to try to.
In mice, we never look at that. We sometimes if we do like lung infection models, we would look at the mediastinal lymph node, which is kind of near the thymus. But I don't know anyone who ever gets thoracic lymph nodes in the mouse. No, there may be people. I don't know anyone.
Sure. I wonder if
you got the ones you can find.
Yeah.
Right.
Do you think the thoracic maybe was larger than the mediastinal in humans? Because I too am used to seeing mediastinal. Maybe the size. Yeah.
I don't know.
Interesting.
I have a question.
So
they make an interesting statement, which I thought one of you could explain. Human control lymph nodes contain germinal centers, possibly because of ongoing adaptive immunity initiated by commensal antigens. And so this means you would not expect to have germinal centers in a healthy, uninfected person. Is that correct?
No, it means that you would. It means that right now all of us are have some germinal centers.
But why do they say possibly? Why do they say possibly?
Why do they say possibly?
So possibly.
Well, they don't know where it's coming from.
Okay.
Whether it's some low-level infection or commensals.
So in other words, at any time, if you sample any of our lymph nodes, you'd find germinal centers, right?
I think depending, they would be maybe underdeveloped or small. They obviously would be, when you feel those lymph nodes, you can see that they're bigger during infection. Yeah. But one of the things they don't tell you, really, there's not very much information about the control people who died. And so that's when you're looking at autopsy patients and you're comparing, maybe they had a viral infection. I mean, we do see a very striking, phenotypic difference in these pictures, but maybe they had germinal centers because they had some underlying infection. Right. I don't know.
Well, we also, I think that probably also goes back to what Steph said about which lymph node you're looking at making a difference. I think that in a healthy person, certain lymph nodes are going to have more germinal centers than others.
I guess respiratory tract would be a good one because you're always inhaling things, right? And,
Right.
Yeah.
Yep.
Yeah, well, I should note that, like, if you're doing human immunology and want to look at germinal centers, the common tissue is human tonsil.
Mm-hmm.
Typically get from kids that have tonsillitis, which they have a lot of infections. Their tonsils are just totally filled with germinal centers.
And I think we'll talk about that a little bit later when they want to look at a certain cytokine in relation to activated status of a lymph node. We'll talk about that in a little bit. But with this figure, the interesting thing they bring up is something called BCL6. And I think I'll let Alex maybe describe what that is. But there's definitely a lack of BCL6 positive B cells. But with that, there's another subset of B cell that is not decreased, which we'll talk about. But I'll let Alex talk about the BCL6 positive cells and what that means.
Okay. Well, BCL6 is a transcription factor that's very near and dear to my heart. So when I was a postdoc, I actually cloned it in Lou Stout's lab from a B cell expression library. And then we found it was Ricardo Della Fava's group showed that it was translocated in B-cell lymphoma and seemed to be an oncogene. So that really inspired us to look at it further. So it's a BCL because it's B-cell lymphoma number 6 gene, but it's a transcription factor. It's a transcriptional repressor protein. It has a so-called pause domain on the N-terminus and zinc fingers on the C-terminus. So I made a knockout mouse for it when I was a postdoc, and the mice had very clear immune deregulation, where spontaneous immune inflammation and early death. They had a growth retardation, and the really prominent thing was a lack of germinal centers. So sort of not unlike what is seen in these poor patients with the COVID, who passed away from COVID. It's definitely, but that was because we knocked out BCL6, the gene, and this is clearly a different phenomenon in these patients. But anyway, BCL6 is a transcription factor that controls development of germinal center B cells. So without BCL6, you can't form germinal centers. And what's interesting about it is you need it in both B cells and T cells. So there's a special class of T cells in the germinal center called these T follicular helper cells that express high levels of BCL6. And if the T cells don't have BCL6, they can't form TFH, T follicular helper cells.
Is there a difference in the phenotype of mice? Have they been made if you knock out BCL6 only in the T follicular helper cell versus in the B cell? Do they have different phenotypes? Because I would presume that it'd be worse if you lack it in the TFH, where you might still be able to get something out of the B cells if they express it, or is there no difference?
That's an interesting question. No one's really, and I've never done this, actually carefully compared them side by side at the same time. I think there are some subtle differences, but like, you can get pretty good antibody responses without germinal centers. It's just you can't get high affinity, certain types of isotype class switching, but particularly high affinity, you have some loss of B cell memory without BCL6 and B cells. And it's sort of a very similar phenotype if you knock out BCL6 and T cells. Yeah, I mean, I'm surprised. I mean, what we did actually early on is made some chimeric mice where we could sort of knock it out in T cells versus B cells. And, you know, I think the overwhelming phenotype is the loss of germinal centers.
Yeah. And,
you know,
and
yes, go ahead.
Oh, I was going to say, I think that's what they're giving us clues in this first figure as to what cell type matters and what this phenotype is that, okay, we're not seeing the BCL6 and we find out there's maybe a lack of these BCL6 positive T follicular helper cells. And so what are they helping? And the name kind of gives it away, the follicle within, the lymph node, and inside the follicle, then you're going to have germinal center development. And these cells are required, for development of the germinal center structure and function, but then of course these high affinity B cells. So how did these become your favorite cell type, Alex? Is it something with the BCL6? I mean, that probably was where it started. I think in 2009, it was discovered that this was the transcription factor for this particular cell type.
Right. Yeah. I mean, when I started my own lab, I took BCL6 knockout mice with me, and like, I kind of agreed with my advisor that he would look at BCL6 and B cells, and I'd look at BCL6 and T cells. He didn't really do that much actually with BCL6 and B cells after that, because he had other things going on. But I stuck with the T cells because I was trying to figure out what it did in T cells. And what we saw is like a really potent development of Th2 type cells in the BCL6 knockout mice. So I was trying to figure out how BCL6 regulated Th2 cells. And then, you know, there was a couple papers showing that BCL6 was in T cells in germinal centers, but we didn't know anything about those cells really. That they were called germinal center T cells and very poorly understood. And it wasn't until people actually showed that when you knock out BCL6, you don't have these cells and you lose germinal centers. That's when the whole TFH field really took off.
That's what I find amazing is that this cell type is, I mean, a lot of these cell types, when there's low levels of them, it's not like they're in huge proportion, but they have these huge function. I think a lot about like innate lymphoid cells. They're always in such low numbers, but they have this huge impact. And so so we're losing BCL6 positive T follicular helper cells, but what's interesting is that they retain something called AID, activation-induced cytidine deaminase. This is a very special enzyme that helps to introduce point mutations in a particular region of the developing B cell so that it will produce antibodies, well, it will produce genes that produce antibodies that are varied, and those are really what is selected in the germinal center for high or low affinity based on positive or negative selection. And so I think what generally they're saying is these AID positive B cells are not lost. So there is still activation happening in or around the germinal centers that they're going to define in a little bit where that activation is happening. But so we're losing BCL6 positive, which is like specific for the memory, but we still have some B cell activation going on. So what is that all about? I think that sums up figure one. And then
I should point out that AID, it actually has two roles. One is it's required for isotype class switching for immunoglobulin and also for somatic hypermutation of the immunoglobulin, which is important for affinity maturation. Probably, so like if you don't have germinal centers, you don't get affinity maturation at all. Probably the AID positive cells are from cells that are undergoing isotype switching, because that can happen outside the germinal center.
Right. That idea of like extra follicular B cell development. So not within the germinal center. Right.
Yeah. Which they talk about later here in this paper as well.
Yeah. So I think their next question was, well, let's take this to another lymph organ, which is going to be the spleen. And they want to see if they can find similar phenotypes within the spleen. And they do. They see the same decrease in B and T cells. They see a decrease in the BCL6 positive B cells, but then you retain the AID positive B cells. So I think looking at that and seeing the similar phenotype, they thought, well, this isn't something that's specific to a particular lymph node, because then you would think about like, something that's retained within that lymph node, that something specific, but this is something outside. This is something that may be inducing systemic effects. And so they thought then, well, let's go ahead and look at cytokines.
Yeah.
Because cytokines, go ahead.
Oh, no. I was just going to say, there were a couple of questions I had when I looked at this figure
Sure.
that I think that later parts of the paper address really nicely.
Sure.
And so one of the things they say is, they see here something that suggests there is an attrition of B and T cell numbers. So they've lost B and T cells here and in the lymph node. And part of me wondered, well, how do we know that we've lost the B and T cells and not that the B and T cells are in the wrong place?
Yep.
What if they're all in the blood and never
Yep.
made it to the lymph node?
Yep.
What if they sort of are mislocalized? And I think that some of their later data addresses that. And so that's a nice thing. But I was a little confused about how they could say it was attrition this early. And they also pointed out here that the AID positive B cells, just like Steph said, were preserved. And so they thought that this probably meant the helper T cells were still contacting B cells.
Yep.
Even if that contact wasn't going quite right.
Yeah. So activated T cells can help B cells, and they don't need to develop into T follicular helper cells. So the TFH cells really are important for the germinal center reaction.
And it seems to be there. So Alex, there's certain signals that would be required for a CD4 T cell that can activate B cells in maybe an extrafollicular space to be able to differentiate into these T follicular helper cells, correct?
Right.
And it seems to be in this paper, that's what they're suggesting. There's the mechanism maybe lies within that. It maybe lies within the inability of these T follicular helper cells to either migrate or develop into the germinal center and cause those interactions that would lead to a very robust B cell response.
Yeah, I was just going to ask, Alex, when we see that kind of extrafollicular interaction as opposed to the interaction in the germinal center, does that lead to a different type of B cell activation, a different phenotype? Is it sort of aberrant B cells in any way?
I mean, it can be. I think they've seen those extrafollicular reactions in like autoimmune diseases and certain types of weird, inflammatory conditions. But I mean, in general, if it's outside the follicle, then it's not quite regulated very well. So, one thing I wanted to mention is if you look in the figures, like the BCL6 stain is really pretty. So like if you just have a section of tonsil or spleen or lymph node and stain it, there we go. Yeah, so if you just stain for BCL6, that'll perfectly line up with all the germinal centers. So like if you just stain for BCL6, the germinal center is all BCL6 positive cells. Now there's other markers we use for germinal centers as well, but BCL6 is like a great marker for BCL6. And so you can clearly see in that one right in the middle, the lower middle, that orange BCL6, there's, that's a really nice BCL6 positive germinal center that's not in the COVID spleen.
Right.
I had one question, and that is, so Vincent asked, if we looked in a normal lymph node, would you, expect to see these germinal centers? And we talked about, what they might be reacting to. The fact that you don't really see any, it's striking me, and I don't know the answer to this. Does it mean that you're not developing them, or could they actually be dissolving? So are there any triggers that might be causing them to fall apart, dissociate, or downregulate something that we would call a marker for that? Although you can do histology and you see that they're I mean, because you can usually tell by the H&E stain just that there's a grouping of cells that's a follicle. It's easier to see, but I don't know. I mean, do you have any thoughts on that?
Well, I think you'd have to do a time course to really figure it out. My guess is that they probably initially tried to form these structures, and then it started to fall apart with all the inflammation. So, like, as the paper showed, maybe I'm jumping ahead, but like inflammatory cytokines can interfere with development of TF8 cells. So,
Yes.
like, maybe they'll start to form them and then they don't fully mature.
What a like perfect segue.
I was just going to say that. That's the
Wow.
very next thing they did.
Yeah. And so they did, because they saw it in both the spleen and the thoracic lymph node, they thought, well, okay, it's something that is systemic that can travel between many different parts of the body. And that would be cytokines. And so as we know, with severe COVID-19 patients, there is an inflammatory signature. And these cytokines we talk about a lot, IL-6, IL-8, so that's interleukin 6 and interleukin 8. They're produced by macrophages, neutrophils, and in addition to that, you have dead and dying epithelial cells that are causing these reactions to happen because of binding of dead pieces of cells. But what they really honed in on was TNF alpha.
Mm-hmm.
I am not particularly sure that I know TNF alpha is one of these acute phase response cytokines, but I don't know if it's necessarily always been correlated like IL-6. So I thought it was interesting they honed in on TNF alpha, other than we know, as Alex said, in a very high level, it can deteriorate germinal centers.
Right. I would agree with you, Steph. I don't think that there has been a lot of previously published literature saying that COVID-19 is related to TNF alpha changes. But they do talk here about other ways where TNF alpha has been related to changes in secondary lymph nodes. So that's more where the tie is, not in the thinking about the clinical TNF alpha seen in patients. One thing that is sort of cool about this, though, which Vincent mentioned before the show, is that there are lots of drugs that are used in some patients with autoimmune diseases that are specifically blocking TNF alpha or the TNF alpha receptor.
Yep.
And so if we were to really nail down that TNF alpha was causing a problem here, perhaps you could imagine thinking about some of those therapeutics. I don't know about timing. I don't know about dosing. I don't know about how you get it in the lungs or any of that, but
yeah, it is interesting that TNF alpha has not gotten much press in the COVID-19 era. It's mostly IL-6, right?
Mm-hmm.
Yeah.
No, I don't know that there's a huge amount of data out there supporting a consistent high level of TNF in all the patients. And it's kind of variable.
And maybe that's because really what they did not, and correct me if I'm wrong, this was not systemic TNF alpha. They looked at, how they looked at it was they pulled out the tonsils, which we talked about before, is this activated lymphoid tissue in both control and COVID-19 patients. And it was this tonsil that had the increased TNF alpha. So maybe it's locally produced that's causing the issue.
Yeah, and people have been doing.
I should point out that, so they didn't really say this, but directly, but IL-6 is actually helps promote TF8 cells. So I think that's why they didn't focus, even though IL-6 is associated very strongly with COVID inflammation.
Yep.
It wouldn't be negative inhibitory for TF8 cells.
That's probably why it would be a little confusing. It maybe wouldn't play nicely with this story. But can you talk about, so with TNF alpha, I mean, we know, I remember reading TNF alpha knockout mice don't develop germinal centers at all in some previous papers I was reading. So it seems to be maybe that it's this too much or too little with cytokines. There's not a clear answer.
I don't remember the literature on, I mean, part of it is TNF alpha is actually important for the organization of the lymphoid follicle. And so, like, if you don't have that structure in place, then you can't form the germinal center.
Right. And so maybe with IL-6, what you were saying is, I mean, obviously it's needed locally for T follicular helper cells, but it's an overwhelming amount in COVID-19 patients. I mean, you don't want that. Right.
Don't want which? I'm sorry. I didn't.
Oh, sorry. Well, so for IL-6, I mean, I think it's just this concept of cytokines are good, but they can be too much doesn't mean better, obviously.
Goldilocks.
Yeah. Yes.
Well, I mean, so one cytokine they didn't talk about, which is really well known to inhibit TFH cells, is IL-2, which is made at high levels in any sort of strong immune response. So, I mean, that would be something I'd be interested in them looking at, but they didn't talk about it. So.
For sure. Definitely TNF alpha was just that it was higher and it fit that story quite nicely.
Yeah, I don't think they gave a really strong rationale for TNF alpha.
I think their rationale was just that they saw things in the spleen as well as the lymph nodes. So that suggested to them that it was some sort of systemic something that was signaling. And so a cytokine would be most likely. I think that was really the predominant rationale that they used.
They also cite some literature saying that in cytokine storm mouse models, you can reverse germinal center loss by blockading TNF alpha.
Yep.
Right. So that's another link of some sort.
Yes. So I think, for people who are curious about, well, how can you really find out? Well, this is where you go in and you have mouse models that you can knock out TNF alpha. You can add it back and see exactly what's happening and also infect them with SARS-CoV-2. So this would require obviously BSL-3.
Yeah, no, as I was going through this paper and I was thinking about kind of what the next study I would love to see would be, I had all sorts of next questions and all of them would require a mouse model.
There's caveats though. Is it this TFH cells? They're not the same in mouse and human, right? They have different requirements for cytokines. Is that correct, or am I wrong?
You're not wrong. They're a little bit different. Yeah, their development's a little bit different. It actually hasn't been studied very systematically, I guess it's a better word. Like it's something I've been interested in because, in vitro. So one reason I never got into TFH cells, or I had trouble finding BCL6 function in T cells, is because if you activate T cells in vitro, they start making a lot of IL-2, which downregulates BCL6. So you could never really see much effect between wild type and knockout T cells. And so IL-2 is really a potent inhibitor of BCL6 and TFH cell development in mouse. And oddly, that really hasn't been shown yet in human cells. Like there's some hints of it, but no one's definitively shown it. And the big difference seems to be that TGF-beta helps promote human TFH cell development, and not so much for mouse. And that's it's sort of like reminiscent of Th17 cells too, because they're kind of different priming conditions between mouse and human, and particularly the role of TGF-beta. But it's sort of reverse. It's weird.
Well, you're good at transitioning to next topics because we're talking about, you're mentioning all these different types of T cells, T helper cells. And so what they wanted to do next was really identify, well, you have this change in the numbers of CD4 T cells, but what are the types that are changing? And so they look at certain transcription factors that define these different T cell subsets. And they identify that it's Th1 CD4 T helper cell that are actually increasing. And these may be then in relation to what you're seeing with the decrease in development of T follicular helper cells. So, Alex, I didn't know if you had any thoughts about that with Th1 kind of increasing in frequency and the role of Th1 cells in T follicular helper cell development.
Yeah, well, there's a lot of papers have looked at kind of like there's this binary developmental pathway for T cells during a viral infection, whether they go into Th1 or TF8 cells. So that's kind of, I guess, if the TFH are blocked, then it's sort of natural that Th1 would increase. So that made sense to me.
Yeah. When I looked at this part of the paper again, the little part of me said, okay, is this a situation where all types of CD4 T cells are inhibited, or is this specific to the T follicular helper cells? They make a lot of arguments about the T follicular helper cells, but they don't do any comparisons until this point with
Right.
other T cell subsets. And so I really liked this figure because it showed that this is a specific decrease in T follicular helper cells. It's not true of all the T cell subsets across the board. And then in fact, that there is this increase in Th1 cells.
Yeah,
Right,
I totally agree. Yeah.
Yeah, and it gives kind of, I think, credence to what Brienne was saying. We don't exactly though know why they're changing, right? We don't know if they are being retained and they're not leaving, and so they're not going to systemic, or a lot of the dynamics of migration of immune cells. Really, with one snapshot of a lymph node, you don't really get at those type of kinetics. So again, we need mouse studies to look at that. But I think I agree. It was nice to see this because you need to know: is it just numbers changing, or is it actually is it specific to that cell type?
Yeah. And the other thing they start doing around this part of the paper is they do get to the conversation we were having earlier about follicular versus extrafollicular cells.
Yep.
And whether these cells are in the wrong place. They actually cite somewhere in here a paper by Woodruff that was a preprint. It's been published since then in Nature Immunology. And it actually describes the extrafollicular B cells here quite nicely. And goes into a lot more detail about what's going on with those extrafollicular B cells. And so they have some parts here where they start talking about how maybe it's mislocalization of these cells and going into these aberrant extrafollicular cells that are related to things you see in disease. And there's some other evidence of that in the literature as well that kind of comes together with this paper nicely.
Yeah, definitely. And so they also describe some other types of T follicular helper cells that change in relation to other types of cells. And Alex, I'm curious, so what does CXCR5 in relation to TFH cells? And I guess either that do or do not express BCL6, what does that marker, how does that play a role here?
Right. Yeah. Well, CXCR5 is one of the key markers used to define TFH cells. So like if you don't, the three most characteristic genes for TFH cells are BCL6, CXCR5, and PD1. Those are most upregulated compared to other T cell subsets. And so CXCR5 is a chemokine receptor that helps the T cell get into the B cell follicle, basically. So B cells normally express CXCR5, or follicular B cells do, but only TFH cells that upregulate CXCR5.
Okay, gotcha. Yeah, I think it's interesting because they do talk about T follicular helper cells that express CXCR5 but don't express BCL6, and kind of what that means in terms of these. Like these TB conjugates.
Sorry, yeah. I mean, CXCR5 can be induced early on after T cell activation, before the T cells become full TFH cells.
Gotcha. Yeah. And I think it lends itself to thinking about the development of these cells, that they're just not necessarily getting the cues to upregulate BCL6. And why that is, they're not going to address that in this paper. And the two things I have is it have to do specifically with a viral protein that actually inhibits something mechanistically, or is it just this, these people passed away. So, obviously there was a hyper-inflammatory response. So is it specific to the virus? I tend to think sometimes not, because they bring up a lot of examples of other viral infections that cause this.
That was my exact experiment, Steph, is I wanted this to then go into a mouse model and to try to figure out which viral proteins were important.
But remember, these viruses that they're citing, SARS, MERS, SARS-CoV-2, Ebola, avian influenza H5N1, these are all, recent zoonotic spillovers, if not immediate, and they're just not. SARS-CoV-2 is I would say is a human virus, but it's still pretty brand new, and it hasn't had time to circulate. So I think these are hallmarks of a virus in the wrong host, and it's probably doing things.
Interesting.
It's doing things as far as a virus can do anything, that are reminiscent of its previous host, which in most of these viruses are the natural host, bats or chickens or whatever. So I think that's part of the situation. And then the well-adapted human pathogens play nicely with the germinal centers, right?
That's actually really interesting. I didn't make that
that's
corollary.
such a good point.
Yeah, that all of the example Marburg, Ebola, H5N1. I mean, these are all spillover events. And right, so yeah, wrong. What did you say? Right virus, wrong host.
Yeah, and
that's interesting.
It's tempting to invoke the common cold coronas, which now are mostly not pathogenic, and I would guess they do not mess with the germinal centers. Oh, maybe they do, and that's why you don't get persistent immunity. I don't know. Someone should look at that,
I don't
right?
know. But there is something. There has to be something different, because even if common cold coronaviruses, which don't mess with the germinal centers, they still don't have great memory B cell responses.
They don't.
Right. And at least these authors, I think, would like us to say, oh, this is the reason for the lack of memory B cell responses.
Right.
I think that's the message they would like, whether, I saw your face there, Steph, whether we go with that or not, and whether this is the reason is another matter. But I think that's part of what they're trying to claim here.
Sure.
And that's why this paper got press.
Yeah, well, right.
Well,
I mean,
it may be part of it, right? But certainly not for common cold coronas. I don't know what's going on there. Nobody's really looked. But.
Oh my gosh, yeah. Don't you wish we would have had like years of in-depth immunology going on with common cold coronas? We had so long.
Well, it's a reflection of what people think is important in the restricted funding that you have. You can't do just anything, right? It's a problem. Right. And now we're in this situation because of that. But now that everyone in the world seems to be working on SARS, Alex, are you working on SARS-CoV-2?
I was a bit. I'm collaborating, but I've kind of scaled back because I feel like there's just too many people doing it.
That's true.
It is hard. I know. Well, so oh, go ahead.
No, sorry. I was just going to say, there's an MD here who I'm collaborating with who's looking at antibody cross-reactivity between SARS-CoV-2 and common cold coronaviruses, and we're looking at.
I mean, has anyone reached out about your BCL6 knockout mice? Do you have BSL-3 access? Because you could start to do some of these.
You got to breed them onto the transgenic first, right?
Right.
Yeah.
Yeah, right.
I thought about doing that, but it's a lot of work.
You mean to get that? You mean to get ACE2 in, Cindy?
Yeah.
Well, you can just use, 2 amino acid changes in spike will allow it to infect normal
the mice
true.
adapted, right?
So you don't. It uses the mouse ACE2, so you don't need to use the transgenic.
Right.
And so you're thinking they might develop serious disease then in the absence of BCL6, right?
Probably, but be interesting to see.
Yeah.
Well, actually, I did want to mention, it's funny that how they looked at these other T cell subsets. So that's, like in a BCL6 knockout mouse, if you just knock out BCL6 in T cells, you see normal Th1, Th2, Th17, Tregs, basically. So it's somewhat reminiscent. It's interesting how restricted the effect is to TF8 cells here.
Yeah, right. Which does make you think. I don't know. That makes me think of something viral. But I think not knowing what's happening with asymptomatic or even during mild infection, I don't know if we'll know. So to
transition to the next part of the paper.
Wow, look at us. We're so good.
Right.
So good at this. I was going to say that, they do start to talk about systemic inflammation, but to wrap up kind of this follicular or extrafollicular section, they also wanted to look at some different types of B cells that they identify by surface marker expression. They call them these double negative B cells because they're negative for something, IgD and CD27 and those markers. It's only just to say that these are B cells that are not undergoing maturation, that they're extrafollicular. They're not, they are class switched, and they probably were induced in a T cell dependent manner, but they're not going to be under selection in the germinal center, which again is this hallmark of memory, of maturation, of affinity. So they show those to be upregulated, or not upregulated, but just increased in these patients.
Yeah, absolutely. And this is where they're starting to actually look at peripheral blood and not just looking at lymph nodes. And so now you can start to see again that this is systemic. And things like, okay, yeah, I'll now completely buy that we have a loss of B cell numbers, because if they're lost in the lymph node and in the blood, then there's not too many places left for them to be.
Yeah.
So
maybe.
So tell me,
Sure.
if you remember a few months ago from the Nussensweig group, they published this study of, they had cloned out antibody genes from a variety of patients, and they were astounded that similar gene sequences were found in multiple patients. Remember that?
Yep.
So how does that in some way relate to these weird B cells getting, not maturing and so forth? If I remember, those were pretty high. Those were pretty high affinity antibodies. So maybe they weren't severe disease patients. Maybe
I was going to say, I don't think that they were patients. I mean, obviously that had died. So I don't know if they were severe disease. And the issue with those, they were high affinity. And I think that the rules that govern so that's a clonal, those are clones, of B cells are different under different selection than maybe what we're talking about here with the migration from like extrafollicular to follicular germinal centers. I don't know the rules that govern that, because I don't know. I think we did a paper one time, remember, when we were trying to identify the proportion between different people who had the same clone of, B cell, and it was so very low. So that's why that paper was confusing to me, because it just seemed to be higher than what I'm used to seeing in papers.
I think the key is they recovered, and they recovered, then their germinal centers are probably fine, right? Or minimally altered.
I would take that paper to say that there is a very specific route that a B cell can take in order to make one of these antibodies. And so most of the B cells who make these antibodies have been selected along that route. But I don't know how much it tells us about what the route is.
Yeah. Okay.
Yeah, that's a good point. So with the systemic, they start to break down their patient population with even more granularity. So if you think about the numbers of people that we had with the lymph node, we had like 4 and 5 patients, which, it's also very qualitative because we're looking at microscopy here. We have more patients. I think in the groups there was like 10 and 15, but they subdivided them by their levels of something called CRP, or C-reactive protein. And that is an inflammatory marker that is elevated during, ARDS, but SARS-CoV-2 in severe patients. And so they wanted to see, do these aberrant B cells, these non-germinal center-derived B cells, do they stratify within like this high, medium, or low CRP level? And they show that they do, although when you say correlate, when using numbers that are as low as they used, I would say associate. I don't think I'd say correlate. Although they do show a regression plot somewhere, but I don't know. So what do we think about this? So Brienne's right that maybe we can correlate what's seen in the lymph node in terms of B cell subsets to what's seen circulating. Because they do see, some of these, they're activated B cells, but they don't express CXCR5. And obviously they're not expressing the BCL, BCL6. So I think it's just suggestive that, I will go back to say, it was a little confusing because at parts they said that these certain B cells they're looking at, they were increased in like a high C-reactive protein group, but in the context of lymphopenia, it's challenging to see those increases, right? Because you actually have the whole population of B cells decreasing.
Yep.
But you're trying to stratify a decreasing subset. That's where I have a challenge. This section was a bit challenging for me to believe, just because the numbers aren't there.
Yeah, well, as Alex said, you have to do a time course, and you're not going to be able to do that in people. So this needs an animal model, right? To look at this. Yeah.
Yeah,
right.
I was very intrigued at one part where they say, based on what they see, B cells either develop very inefficiently in the bone marrow or are possibly generated but acquire an activated phenotype and are lost. And so the idea that the B cells were developing just fine, but then they were going too fast afterwards and skipping steps was an interesting idea to me that I hadn't really thought about before.
Yeah.
I don't know enough about all of these B cell subpopulations to say whether I can totally buy that, but I thought it was an interesting idea.
Yeah. Alex, do you see, when have you looked at, germinal center development and tried to correlate it with systemic, I guess we would say biomarkers. Some people don't like that word, but systemic phenotypes of B cells and T cells?
Short answer, no. But I mean, that's an interesting question, but no, we haven't really done that. Sorry.
No, it's fine. I think that's what they're trying to get at here, is to see if what they, saw in the germinal center is similar to the systemic. And if, they talk a lot about like these transitional B cells called T1 and T2. And these transitional B cells are moving out of the bone marrow. They're getting cues to exit and then migrate towards, lymph nodes to then be interacting with antigen presenting cells and have these cues so they can transition, but they're stopped. It seems like they're halted before they can really continue on with this maturation process. So in the grand scheme of things, I think the big picture that this group is trying to show us is that, you just have a lack of development of the germinal center because these cells are either not migrating there or they're not getting the cues to maturate there. And when you don't have a certain cell type, the whole structure of the germinal center can deteriorate.
Yeah, but I really doubt that that's why they're not getting germinal centers. I mean, maybe.
Okay.
I mean, I think it's just there's multiple different things going on. Like this figure, I felt like it didn't really fit in with the rest of the.
Which one are you mentioning?
Sorry, figure 5 with
the
small B cells. I mean, clearly there's some dramatic loss of B cells with severe disease, but
right. This is just like subsets are changing in the systemic. Like that's what we're seeing.
I mean, I certainly don't think there's evidence that the B cells aren't developing in the context of this infection, and that's why you're not getting germinal centers.
Right. Like we would definitely need some additional mechanistic studies to be able to say that. Yeah. Right. All we can really say is that the germinal centers are gone, BCL6, T follicular helper cell development is lower, but by just looking at phenotype changes in the blood in like 10 people, we're not going to know why.
I got to say that they say their last sentence of the results is these data establish that the germinal centers go and then these weird B cells accumulate in the blood. They think it's...
I mean, okay, we're establishing with like 10 patients. Okay. Great. Yeah. So, and what do we all think about the fact that we're using people who were severely ill and died?
I feel a little better that they have the data from the peripheral blood where they can include some patients who had less severe disease, but it is really hard at the beginning to know how much of what they're seeing is severe disease and whether you would see something similar in other patients.
Yeah.
I mean, it would be really interesting if this only happens in fatal COVID, right? Which would then beg the question is why doesn't it happen in others? Do they control the infection and hence there's less of an inflammatory response? It's interesting questions, right?
Yeah.
What would we measure though? Because I don't think you're going to be getting lymph node biopsies.
No, you could get tonsils. You could get tonsils. They're in the right place, right? They're back there.
Yes, true.
You could.
Would that be enough? Would we be able to sample tonsils from mildly infected people and gain information from that? Do you think, Alex?
Maybe. Actually, I don't know anyone who's ever done that, but I do know they can do microbiopsies of like puncture biopsies of lymph nodes. So you don't need to take out the whole lymph node. You can just get a sample of cells, and like Shane Karate's done some of that. They've done a lot more with like macaques, I think, because those animals are so valuable, so they want to be able to assess their immune responses while not sacrifice them.
Unfortunately, but macaques don't get severely ill, though. That's the problem, right? They just have a mild disease. So there is a mouse model made by Ralph Barrick where the virus was passaged in mice and it acquired changes that make it cause acute lung injury. And I'm sure they're looking at this germinal center issue there, but it may not do it, in which case you can't say that disproves it because they're mice, right?
Yeah.
They're mice.
So I was wondering, because I saw today there was information about a UK study where they're going to challenge humans with SARS-CoV-2. And I wondered whether, that's a whole nother issue, right? Because I thought, well, we can come back to that. But yeah, so I thought, my first thought were some of these questions is if you could do those microbiopsies or you could do something like that, you could learn a lot from those people. I'm assuming they're going to get a fair amount of money to do this, and they're going to volunteer to get many, many different samples collected. I don't know.
I think these authors would argue that you can look in the blood and that's enough. Our study shows that it's all you need to do.
Yeah.
You look at those overlapping error bars, it just seems to be that there's quite I know they saw significant differences in these subsets in the severe versus convalescent versus, when they did low, medium, and high CRP, but there are some people that had very high, let's say these double negative B cells that were even in the convalescent. So it's hard. Yeah.
And doing flow, those gates, just
I learned from you. I learned that from you, Cindy,
because you completely different.
You pointed that out multiple times in papers.
Alex, you were going to say something maybe before?
I'm sorry, I forgot what.
It's fine. It's fine. So there is a last part to this paper that they kind of just they wanted to throw in there. And it was, they're looking at B cell subsets, but are they looking at, is this a SARS-2 specific B cell / T cell effect, or is this just a general broad phenotype? And so they showed that, it bound to the RBD, which is the receptor binding domain and the spike protein on the surface of SARS-CoV-2. They only looked at RBD. They didn't really do, which maybe it wouldn't be necessary to see all the different proteins on the virus that it binds to. But they kind of said, well, it binds to it. And they kind of gave themselves the caveat, like, this is only one, protein. This isn't strong evidence, but we wanted to show you it was specific.
Yeah. And so the idea here is they want to say this isn't some global B cell change. This is a specific change on the SARS-CoV-2 specific B cells. And, in theory, that could lead to the idea that this might be a virus specific type of effect. Though, of course, I think there might be a lot of other things that you might want to do here to show what's going on.
Yep.
Right. And really, I guess you could have gone all the way from the beginning and maybe tried to define SARS-2 specificity with, staining. You can stain for that in some of the lymph node biopsies. And for sure in the peripheral blood, you would definitely have enough to be able to look at that. So
I don't know, can you use that tool? So they use a really neat tool, right? They double label the recombinant receptor binding domain, and then use that to look for B cells that have antibodies that recognize that, right? Can you really stain tissue sections with that? I suppose possibly, but I'm not sure.
No,
once you fix and do antigen capture and all of that, I don't know how well those
that you would actually get. Yeah, the sensitivity of that by the time on a fixed tissue.
No
That's
idea.
a good point.
Yeah, I don't know anyone who's done that.
Right.
It'd be technically very challenging.
Very challenging.
Yeah, it would.
And do you think that they probably didn't use flow cytometry for the lymph node samples just because they only had so much?
I think it's probably because they only had so much, and because with the lymph node, the actual architecture of seeing that not only that the B cells are present or absent, but that they're in the right place is so important, that they went for this microscopy.
They also may have gotten fixed chunks of spleen or lymph node. They may not have gotten fresh to be able to disaggregate it into flow.
Okay, that makes sense. Yeah, I'm sure there's a lot of people, vying for these samples from.
Oh, yeah.
So I'm sure, this group got this chunk and another group got another chunk. So.
Yep.
So that, kind of wraps up this paper. And I guess as we kind of rounding out our episode, what are some things that we want to talk about? I mean, there's obvious implications for memory development and vaccines.
They make
And
this.
I guess-
they make
yeah.
this statement that this has implications for the development of herd immunity by natural infection, right? And as we've said, they've only looked at people who died, right?
I think we need to
Right.
look at
And that's
not people who died.
Yeah, I mean, there's
Well,
no.
I don't exactly understand the statement. I mean, how would that lead to herd immunity?
Well, they're
That's
saying
an
that
individual.
basically, and I think they're saying in everyone that we must be making crappy antibodies because of the destruction of the germinal centers, and there's no memory and long-term immunity. But I don't think that's at all established from this paper, right?
Mm-mm.
I think there are other reasons why natural infection herd immunity is a bad idea. Also, I don't think we
Well,
need
yes, for sure.
the take home from this paper to tell us that.
No, I would like to have that as ammunition to tell these people, stop saying we should let the virus rip through the world, right? But this is not it,
Right.
unfortunately. But
I think what I took away from it though was that if this is a specific effect
Mhm.
of the viral infection that's inhibiting or blocking or disaggregating or whatever it's doing to the proper development of germinal centers and the production of class switched high affinity antibodies that are protective, then, and it's happening during infection and it might not happen during immunization, then that would be an argument for natural infection being worse than vaccination or immunization, right?
Right.
Oh, it's
Exactly.
already not a good idea because so many people die, right?
Right.
Natural infection is not a good idea, even though 80% are mild, 20% are serious. So,
Right.
but I agree that maybe a vaccine will not lead to the cytokine storming and the TNF, whatever, overproduction that trashes the germinal center. So, yeah, but the vaccines also have to work, right? They have to
That's true.
do
They do,
protective,
they do. And-
long-lasting antibodies.
Yeah, go ahead, Alex.
Well, they cited a really interesting paper though in here. It's from Dan Baruch's lab, where they in a mouse model they vaccinated mice with the epitope of a virus that only induced CD4 T cells, and then to see what kind of protection that would give, and then when they infected the mice with the LCMV, which is the virus that they immunize against the epitope, the mice got fatal inflammation, like a deregulated CD4 response, and mostly died, which is really interesting. So it's very reminiscent of what happens with some COVID patients, that there's some deregulation of the CD4 T cells and the B cells, and like they talk about in this Shane Crotty paper, like there's not coordination between the memory B cells and the memory T cells.
Right.
That they need.
And maybe that has to do kind of what this paper is saying. The synergy between B and T cells also has to happen at the right spot, because happening in the wrong location, so maybe not in the germinal centers, could lead to a buildup of a phenotype that is going to produce antibodies that maybe cause, I'm just throwing that. I'm not saying they cause it, but like antibody dependent enhancement or some other type of aberrant response that we would not want with a vaccine or a natural infection. And yeah, that's interesting, because I think in vaccinology, it's interesting. I think in the past, the field has been, if it works, that's great. I don't know if we always investigated the mechanisms of why vaccines work.
Yes, exactly.
Right. And now we're kind of left with really, we're at a point where the field of developing vaccines, we can get very specific with the type of antigen and the delivery method. Is it mRNA or DNA? Is it protein? And we don't have the answers to those questions as to, well, what's going to happen in the germinal center? And so we're at the stopgap of like lack of information, but we're in a pandemic and we need a vaccine now.
It's totally
very
empirical. It's totally empirical, right? We make this vaccine and we try it and see if it works.
Yep.
That's
Yeah.
all there is. It can be an mRNA, it can be a vector, it can be inactivated. It doesn't matter. We're just going to see what works. And there's no real rationale, except most of them are focused on spike, which may or may not be the right thing to do. We'll see.
Sure.
It's sad because, I mean, the one good thing is it's all happening very quickly, right? Maybe in a year and a half we'll have a vaccine, but on the other hand, may not be the right one initially. Who knows?
Sure. And maybe there's a vaccine that induces more durable antibodies than another, but we actually won't know that for,
Yeah, because
while.
there's no
Time.
systematic way of doing that, right? We don't have any clue.
Yeah.
In part because this is a new virus. But as you say, even in all previous history of vaccinology, we really don't know what makes a good one. Like the new shingles vaccine, right? Shingrix. It's the single glycoprotein adjuvant, and it's amazing. But no one would have ever predicted that, right?
Right.
All the more reason why sort of basic immunology and understanding immune memory and understanding the germinal center reaction and all those things are so important.
Yes. So Alex needs to keep studying the germinal center and BCL6. Now, you study this interaction in the context of allergy and IgE. Is that correct?
Lately, yeah, that's
Okay. So I guess, do you have any like tidbits of info in regards to how SARS-CoV-2 could be interacting in the context of somebody with allergy or hyper IgE? I know that that's a pathology that people are worried about, like inducing hyper IgE with a vaccine, let's say.
Yeah, I mean, I know.
I know these are big questions, so it's okay that you don't know.
No, I know. I don't have any great insight. I know that like asthma is a comorbidity for and SARS infection, COVID-2 infection, and if you're making a Th2 response, which if you're more Th2 prone, that's probably not as effective as fighting the virus. It doesn't induce the right type of antibodies. Yeah, I mean, I haven't really. Actually, one thing we're really interested in right now is Iga regulation, which is the secreted antibody
It's my favorite.
made in the lungs. And there's very little known about how that's regulated in vivo, or particularly how it can be controlled for a vaccine.
So one thing that they do here is they look, in things like the thoracic lymph node. But I could also imagine looking at things like NALTs or BALTs and sort of the follicles that are being made there could be important. Are people able to look at organized lymphoid structures in those tissues? And do you see BCL6 positive cells there?
In GALT? Certainly in GALT, yeah. I mean, well, Peyer's patches, of course, if you define Peyer's patches as GALTs. There's a lot of germinal centers there. I mean, in fact, they have very high constitutive levels because of commensal antigens.
And for those, the uninitiated, GALT is gut-associated lymphoid tissue. Oh, no, it's fine.
Yeah, NALT is
nose, nasal.
I don't know much about
BALT is bronchiolar.
Like
but in the lungs, though, there's like organized little patches, but they're not Peyer's patches, and they're not the same as in any other organ, as far as I understand.
Those are weird. So those are those inducible BALTs.
Yeah.
And those are very different. So like, did these patients who died, do they have inducible lymph node? What is it? It's bronchial associated.
Bronchiolar associated lymphoid tissue.
So it's BALT. That's just what it is. It's BALT.
Yeah, so you'd have to look at their lungs to see if there was any eyeball in those patients.
Yeah, but aren't they like inducible lymphoid follicles?
Yeah,
something like that. But yeah, so we've been talking about the generation of antibody responses in germinal centers within lymph nodes or within the spleen, but then you also have these patches where that can happen, right? So we really don't know whether those responses are normal or abnormal, unless they take lung biopsies and do that.
Right.
Doesn't HIV trash germinal centers too?
It does, although I think of other lymphoid organs getting trashed first, but that's probably because of places I've worked.
Because I know that
depletes everything in the gut, right?
Well, in the gut, there's no more GALT left. If you look,
it messes up the GALT, it messes up the thymus. It does also mess up the germinal center.
Yeah, because
actually, in latent infection, I think it only trashes T cells in later, the severe later stages of AIDS. Actually, so it's interesting that TFH cells are thought to be a reservoir for latent HIV.
Well, they express. We said they express CCR5 or no?
CXCR5.
CXCR5.
They do express CXCR4, which I think that's one of the co-receptors for
Yes.
HIV. So,
Yeah.
Yeah. So a lot of implications, even for you thinking about co-infections, things that many years worth of work that we have ahead of us. So as we finish up, I guess we got like 5 minutes, maybe till the end of the hour and a half. What are some last thoughts? I think we're going to have to do an all email episode because it's very hard to get to the emails.
Yeah, we'll have to
papers
do
like
that
this.
next time because we don't have any time left. But
Right.
I just like this paper. It makes a lot of sense, although I think the caveat is that it's in very sick people, and I'd love to know what happens in others. But I think why these people get to this point is really interesting, right? They must have some issue initially, really early, controlling replication. Then it gets out of hand, and then you overreact. And, that's where some of the SNP mapping is maybe shed some light on it. Who knows? But I think that's really interesting. And they note that that's an interesting and very hard question to answer.
Yeah, I mean, I like that, we've been talking and people can argue quite a bit about what's going on with duration of antibody responses, but they actually take that idea a step further and start to look at what might be going on in terms of generation of those responses, and at least give us sort of an idea of a mechanism or an idea of a hypothesis of what we need to be looking at in terms of what might be going on with that duration.
And it really is not the virus. It's nothing specific, right? It's the host that...
It doesn't seem to be. Yeah, it seems to be the immune response. So
yeah, I was waiting for them to blame the virus, but they never did. I mean, of course, it's to blame because it started the whole thing. But if 80% of people can handle this, then it's got to be some host thing that's leading to high uncontrolled reproduction. Anyway, I think I really enjoyed it. I thought it was good, and appreciate Alex's color commentary.
Yes.
All right.
Thank you.
That is Immune 36. You can find all the show notes at microbe.tv/immune. If you want to send us questions and comments, we'll eventually answer yours. Immune@microbe.tv. And if you like what we do, consider supporting us. You are our only means of support, or mostly our only means of support. Microbe.tv/contribute. Our guest today from Indiana University School of Medicine, Alex Dent. Thanks, Alex, for joining us.
Thank you very much.
Appreciate your time. Cindy Leifer's at Cornell University over on Twitter. Cindy Leifer. Thanks, Cindy.
Thank you. Yeah.
What's your Twitter handle, Alex?
I think it's Alexander Dent 6, I think.
Wow, there's 6 of
we'll
you.
Put it
out
in the
there.
show notes.
6 Alexander Dents out there. Cindy Leifer. I said Cindy. Steph Langle's at Duke University. Stephanie Langle on the Twitter. Thanks, Steph.
Yes. Sure. Thank you. And thanks for being flexible with baby Graham in the
No
beginning.
problem.
Appreciate
No
it.
worries. We'll see in 20 years whether she agrees with what you said,
Yeah, well, we'll have to see.
She's going to be an immunologist.
I would let him decide whatever he wants to be, but no, it's fine. But I imagine that, he's going to be around a lot of science over the next X number of years. So we'll have to see.
I defaulted to calling it a girl because I forgot. And, I always default to guys, and Kathy Spindler always gives me hell. How do you know it's not a girl? So I'm now
good.
in
Good you have Kathy
the
in your
girl
life.
default mode. There you go. Brienne Barker's at Drew University. BioProf Barker on the Twitter. Thanks, Brienne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.